quantitative determination of nucleic acids from the samples. This experiment is focused on quantitative determination of DNA and RNA concentrations from biological samples. So we are going to perform these experiments one by one because the procedures are very similar, and both procedures are based on the chemical reaction of sugar component of nucleic acids, meaning deoxyribose for DNA and ribose for RNA. Sugar component in acidic medium at high temperature reacts with aromatic compounds such as diphenylamine with the ribose and orsinol with deoxyribose, producing colored complexes, blue for DNA determination and green for RNA determination. So we are going to take 3 test tubes and mark them as T for test, S for standard, and B for blank. And these are going to be 3 test tubes for DNA determination. We're going to take 3 more test tubes and mark them again with T, with S, and B for the RNA determination. Now we need to pipette 1 milliliter of DNA extract, which is going to be our sample, to the T test tube. Now we need to pipette the standard solution of DNA, also 1 milliliter. And water, 1 milliliter in blank. Now, we need to pipette the so-called Burton reagent, which is basically diphenylamine reagent, 2 milliliters in each test tube. Mix the test tubes and gently put stoppers and put the test tubes in boiling water bath for 10 minutes. For determination of RNA, we should take the RNA sample and pipette 1 milliliter to T test tube. Now the standard solution of RNA, we should pipette to standard. And distilled water. One milliliter in blank. Now we need to pipette 2 milliliters of arsenal reagent to each of those test tubes. Mix the test tubes and put them to boiling water bath for 10 minutes. After boiling and then cooling down the test tubes to room temperature, we can notice the formation of a blue-colored complex for the determination of DNA concentration and formation of green-colored complex for RNA determination. Now we can read the absorbances using photometer and red filter. Absorbances are read using the metal photometer and using the red filter. We're going to prepare the instrument to measure the absorbances. I'm reminding you the upper scale. And first we are going to determine the DNA concentration. Pour the blank solution to the cuvette, put the cuvette to photometer, and adjust the zero of the instrument. Take another cuvette and pour the test solution in it. Replace the cuvettes and read the absorbance. Put back the blank solution. Take another cuvette and pour the standard solution. Replace the cuvettes and read the absorbance. For measuring the RNA concentration and absorbances, we are also going to use the same red filter and the very same procedure. First, we are going to take the blank solution and place it to photometer. Adjust the instrument to 0. If the color of the blank solution is too strong, sometimes you will not be able to adjust the instrument to 0, like in this case. When you're measuring absorbances in such cases, you will have to subtract this 0 absorbance from the rest of the results. So remember this absorbance and subtract that value from other absorbances that we are going to measure. Now take another cuvette, pour the test solution and put it and measure the absorbance. And finally, take the standard solution place it to photometer and read the absorbance. After subtracting the 0 absorbance, use the equation provided in the manual and calculate the concentration of RNA.